Today we're covering Chapter 10, which is going to be our last chapter for Biology 101. So we're going into mechanisms and evidence of evolution. And we're going to start off by talking about a scientist, Wilhelm Weinberg. He's going to be a German physician that's born in 1862 and died in 1937. He pursued medicine at the universities of Tübingen, Berlin, and Munich, and obtained a degree in 1866. After gaining clinical experience around Germany, he's going to return to Stuttgart in 1889 to open his own obstetrics and gynecological practice, and he's going to run this clinic for over 40 years. In his spare time, he maintained interest in human and medical genetics, which he led to 160 publications and recognition of the ascertainment bias. Now, he's going to propose the genetic equilibrium principle in 1908. That's an idea that originally was discredited by scientific community. His contribution to the principle remained obscure as it was overshadowed by similar premise by G.H. Hardy months later. So since recognition of his contributions to the genetic equilibrium principle in 1943, his name remains associated with what is known as the Hardy-Weinberg principle. So what is evolution? Well, evolution is going to be described as the change in allele frequencies of a population over time. A population is going to be a group of interbreeding individuals that live in the same area and can produce fertile offspring. That means that the offspring can reproduce. Population genetics is going to be the study of allele frequencies within the gene pool of a population to determine how these alleles are going to be inherited and how they influence genetic variation. Allele frequencies are going to be the change through mutations, genetic drift, gene flow, or natural selection. So going into each of these changes of allele frequencies, first we have the mutation. The mutation is going to be a change in allele frequencies in a population as DNA base pairs are going to be altered, either converting one allele to another or producing a new allele altogether. Genetic drift is going to be where influences change in allele frequencies due to random chance events. And so here you can see we had a random chance event and the allele frequency changed from more darker rabbits or darker rabbits with some light rabbits to more darker rabbits to the third generation being all of the darker rabbits. Now, with this one, we're going to have the genetic drift occurring in two ways. And so first we have the bottleneck effect. It's going to occur when changes in allele frequencies result in a severe reduction in the number of individuals of a population due to catastrophic events, such as an earthquake or habitat destruction. It could be a volcanic eruption. It could be a tsunami. It could be different things that come in. The second way is called the founder effect. That's where changes of the allele frequency when a small number of individuals from the original population will leave or is geographically going to be isolated due to some event. With this one, gene flow, it's going to influence allele frequencies as individuals introduce their alleles as they migrate between populations. So we see one type of beetle on the left-hand side. We see another type of beetle on the right. The right hand had a beetle that went over to the left, and so it's starting to introduce its new allele frequencies there. Natural selection is going to be changes in allele frequencies in populations as individuals with alleles more favorable to a particular environment have higher reproductive rates than individuals with a less favorable allele. And so here you can come in and you can see the heritable variation that you see of color. So we have the brown, we have the green, and we have the bluish purple here. They're going to have competition for resources. So the competition is going to be eliminated within a population where individuals with those favorable variations are going to be able to outcompete and get more food, more space, and be able to reproduce more in this area. Adaptation, that's where they're able to go in with those traits. They allow that organism to be better suited for the environment. So the organisms with that one, they can be able to actually see some of these. The ones that are able to hide or camouflage themselves are going to be the ones that survive. And differential reproductive success and fitness here, you can see, is where individuals with a beneficial variation compared with others with less favorable variations are going to be able to produce more offspring. And if you produce more offspring, you are considered to be more fit. So here we're going through just the basic definitions here. You saw the visuals on the last slide. So heritable variation, we have that natural selection working on individual variations within a population that are going to arise through mutations and are inherited in subsequent generations. Competition for resources. That's where resource becomes limited within a population, and those individuals with favorable variations are going to be capable of outcompeting others, and they will survive and reproduce. With the adaptations, these traits are going to allow organisms to be better suited to their environment. So that might be camouflage, so that they can blend in and not be eaten by predators, or maybe they're able to outrun the predators, or maybe they can become the predator themselves. In differential reproductive success and fitness, fitness is going to be considered the measure of reproductive success that an individual with beneficial variations compared to individuals with less favorable variations. Fitness is going to influence sexual selection as mates choose their partners based on traits that imply higher fitness, and you're going to have two types of sexual selection. Those include intrasexual, direct competition is what that means, or intersexual, where there's female choice in that selection. So overall, the fitness of an organism is going to be dependent on how it best adapts to its environment. Here we have Crypsis. This is going to be an adaptation in which the coat color of the Pyromyscus pollionotus mice is going to be matched to its environment. And this is going to be one of the most common studied examples of fitness and natural selection. So the old field mice started out as a brown color that's going to match the soil, and it's going to match the grasses and the plants that are going to be in that area. So he's able to camouflage himself, hide, and that makes him able to reproduce and survive to have offspring that then can reproduce. Over here on the right-hand side, we see the Santa Rosa Island beach mouse. This mouse, if it remained the color of the old field mouse, would be easily visible and be able to be picked out by predators. It's adapted. Natural selection has led to its ability for these mice to be able to blend in with their environment. Therefore, they're able to escape their predators. The ones that were darker did not survive, and therefore now the main one is going to be this lighter color mouse. Now we're looking at inter and intrasexual. So looking over to the left with intrasexual, that's where there's going to be competition directly between males to mate. So we see these elk here that are fighting, and the one that's going to win the fight will be the one that wins the ability to be able to mate with the females, therefore spreading his genes and his genetics, and so that makes him more successful. Now, intersexual is going to be on the right. That's where the females are going to be able to choose her mate based on elaborate phenotypic characteristics. So here, the female is checking out the male. The males are going to be the ones that have the more beautiful, brightly colored feathers that you can see. And so they're going to be able to go and they're going to determine who they want to mate with. And so the male puts on the show. Now, natural selection is going to favor phenotypes in three ways. First, you have your directional selection that's going to favor one extreme phenotype over another, and that's going to result in a shift of individuals with that particular phenotype. So here we show different moths. We have one with the original phenotype. That's going to be kind of a spotty, lighter color. And then the population after natural selection is going to go towards the darker moth. So something came about that caused the moths that had the lighter coloring to start to die out. They couldn't compete. Diversity or disruptive selection is going to favor individuals with both extreme phenotypes over an intermediate phenotype. So in this one, we have three different. We have the dark, we have the light, and we have kind of a mixed between the dark and the light. And so here, your original population was the light one. And so you're going to see either extreme, and you're not going to go with the intermediate phenotype. Stabilizing selection is going to favor individuals with intermediate phenotypes over those with extreme phenotypes. And so here you see you have a population before natural selection and then the population after natural selection. So natural selection doesn't always produce perfectly adapted organisms for several different reasons. One, natural selection only works on pre-existing variations within a population. Secondly, the fast-changing environment doesn't always give natural selection time to operate. Third, natural selection does not produce a new anatomical structure, but rather modifies existing structures based on required adaptation. And fourth, evolutionary change is going to be restricted as certain trait adaptations may require a trade-off with another trait. So in with this Hardy-Weinberg principle, we're going to have G.H. Hardy and Wilhelm Weinberg independently proposing a mathematical model known as the Hardy-Weinberg principle. This is going to establish a theoretical state of genetic equilibrium where the allele frequencies within a large randomly mating population can remain unchanged from one generation to the next. So with the Hardy-Weinberg principle, you're going to have allele frequencies using the formula P plus Q equals 1. P is going to represent the dominant allele. Q is going to represent the recessive allele. So example is going to be a population of mice have either brown fur color, which is going to be dominant or big B, or the recessive white fur color, which is going to be lowercase b. The frequency of the dominant brown fur color allele is going to be P equals 0.6. Then one can easily determine that the frequency of the recessive allele by using the formula 1 minus P is going to be Q, or 0.4. Therefore, white fur color allele frequency is going to be 0.4 in the mouse population. You can change this over into percentages by saying the brown fur color is 60% and the white fur color is going to be 40%. Genotype frequencies are going to use the formula P squared plus 2PQ plus Q squared equals 1. So P squared is represented the homozygous dominant allele. 2PQ is going to represent the heterozygous alleles. Q squared is going to represent the homozygous recessive alleles. So the example with this, you can use the mice as an example, since P represents the frequency of the dominant allele. The frequency of the homozygous dominant genotype, capital D in a population of P squared, is going to equal 0.36. So you take 0.6 times 0.6. Individuals of a population that were homozygous recessive, or 2 little d's, is going to be Q squared, or 0.4 times 0.4, which gives them 0.16. Hardy-Weinberg represented heterozygous individuals as the 2PQ. Therefore, these individuals having one dominant and one recessive allele and receiving one allele or the other two ways, either from the mother or the father. So the mouse example is going to be 2PQ, which is going to be 2 times 0.6 and 0.4 times 0.4, and that's going to give 0.48. So when the frequencies of genotypes are added together, they all equal 1. So you can look at it as 36%, 48%, and 16% would equal to 100% if you're changing it over to percentages. So in addition to the genotype frequencies, also represent the percentages as I just described. So Hardy-Weinberg equilibrium has 5 conditions that must be met. 1, no random mutations. 2, random mating. 3, no gene flow. 4, large population size. And 5, no natural selection. So any deviation from these conditions causes allele frequencies to change. Allele frequencies are going to be constantly changing. Therefore, it's rare for a population to maintain the Hardy-Weinberg equilibrium. Conditions are useful in determining whether evolution is occurring within a population. Lines of evidence that support evolution. The first one that we're going to go with is going to be a very strong one, and that's going to be fossil records. These are going to be collections of evidence that provide a look into history of an organism's life. It includes the basic structure, relatedness to other organisms, habitat, and global events that influence the extinction or evolution. Now, extinction means that an animal is no longer with us. If an animal is extant, then that means it is still living. So extant is living, extinct is non-living. So fossils are going to be organismal remains, such as bones, teeth. We could have imprints, and they're located within the Earth's different stratified rock layers that paleontologists use to construct an organism's fossil record. Formations of fossils are going to be rare given the limited number of structures that can produce fossils or the inability of some organisms to leave fossils. Therefore, the fossil record is going to be considered incomplete. Analysis of fossil records has revealed progressive intermediate stages, suggesting the accumulation of changes over time that supports descent with modification ideas. Transitional fossils are going to be a group of fossils that are considered intermediate of the evolutionary change. Common examples of transition fossils will include dinosaurs and birds, Tiktaalik, which are going to be the fish and land animals, the Psammora, which are going to be amphibians and reptiles, and the Therapsids, which are going to be your reptiles and mammals. Most complete, well-preserved fossil records consisting of several transitional fossils revealing subtle anatomical modifications to change environments. This is going to be an evolution of the modern-day horse, from its dog-like, forest-dwelling ancestor to its current grassland grazing genus Equus. So looking here at this fossil record, we could see on top, that's going to be the most modern form. We have our modern horse. And as you go through and look at the different fossil records found in the different layers of Earth, you can see how it changed. As you go deeper and deeper, you can see how it became smaller and how those fossils looked quite different. Biogeography is going to be the study of the distribution pattern of organisms that are living on Earth. We have a unique distribution pattern of organisms that we observe today, and we can explain this by the theory of continental drift. The theory of continental drift states that all continents were at one time a large landmass called Pangaea, which broke apart and shifted their current locations. As one landmass, Pangaea enabled organisms to travel between different regions. However, when continental drift began, it started to restrict movement, and this isolated organisms to specific continents and regions. A common example of how continental drift influenced by biogeography can be observed in diversity of mammalian species that are found in Australia and South America, which were once connected when Pangaea existed. So Australia is going to be home to the most prevalent and diverse species of marsupials that are found on Earth in comparison to placental mammals, while placental mammals greatly outnumber marsupials in South America. Also, it's used to develop fossil records for certain organisms to determine their evolutionary history, as was the case of the genus Equus, which includes the modern-day horse. So here is a look of how Pangaea was put together, and you can see different migration patterns that existed before it broke apart. And this is how these different organisms, both flora and fauna, so both animals and plants, were able to make their way across different regions. We also look at comparative anatomy and embryology when we look at evidence for evolution. Comparative anatomy is going to be the study of anatomical similarities and differences that are among living organisms. Two types of anatomical structures studied using comparative anatomy will be the first, homologous structures. This is anatomically similar but functionally dissimilar structures that are going to be found in related organisms who share a common ancestor. So we have anatomical arrangement of bones and vertebrate forelimbs. So here you see a human. Beside the human, you see a dog. Beside the dog, you see a bird. And beside the bird, you see a whale. We all have the same bones there, but they're going to be used for different things. So therefore, they are similar, but functionally dissimilar or functionally different. Homologous structures, however, should not be confused with analogous structures. These are structures whose origins didn't result from a common ancestor, but independently evolved due to convergent evolution, by which different organisms living in the same habitat adapt to the same environment changes. This results in development of similarly functioning structures. For example, the wings of a bat and a bird are going to be homologous. However, the wings of an insect are going to be analogous to the structures. Although the wings of three organisms appear superficially similar and perform the same overall function, the wings of bats and birds will consist of four-limb bones that are modified by natural selection, while the butterfly wings are going to be extensions of its body's cuticle. The next type of anatomical structure that we study is going to be referred to as a vestigial structure. These structures are going to have their function that is reduced or non-existent, but it was once functional in and inherited from a common ancestor. So an example of this would be the presence of vestigial pelvic and leg bones that are going to be found in snakes and whales. Now these animals no longer have legs, no longer need a pelvis, but those bones are still going to be in there, though very small. Embryology is going to be the study of embryos to determine similarities and differences in structures between organisms at various developmental stages. Work of scientists like Carl von Baer and Ernest Haeckel are going to provide early evidence of similar developmental patterns among different organisms. Many similar features are going to be shared among different organisms during early embryonic development, including gill pouches and postanal tails. But certain genes are going to influence the future development of these structures. And so here you can look at the different posters up here. You'll see the different animals. So going in with F, that's going to be a fish. A is going to be a salamander. T is a turtle. And H is a chicken. Coming over to the next poster or diagram, you're going to see S is the pig. R is the cow, K is the rabbit, and M is the human. Now going through these different structures, notice how the top row all look similar. As they start to develop, you start to see some differences starting to form, especially in the first group. And then by the time you get to the bottom, you see how they develop differently. But first starting off as embryos, they all have similar structures and features, and it would be hard to tell the difference between these different organisms. Molecular biology is going to be the study of the structure and activity of different molecules found in cells, including things like DNA, RNA, and their proteins. DNA is going to be composed of nucleotide sequences which define an organism's genetic information. Organisms that are closely related are going to show minimal differences in their DNA nucleotide sequences. We have laboratory techniques such as DNA hybridization, and DNA sequencing that are going to be used to compare the DNA nucleotide sequences of two organisms to determine their relatedness. Both techniques are going to have revealed that humans are going to be more closely related to chimpanzees than any other primate species. RNA strands are going to be known as mRNA, as an intermediary molecule that's produced by DNA to produce proteins. mRNA is going to consist of 3-letter codons that are translated into one of 20 amino acid based on genetic codes to produce a protein. All organisms have DNA, therefore all organisms use the same genetic code and amino acids to synthesize proteins. Slight similarities and differences in these amino acid sequences of the proteins can reveal the duration of evolution between species, beginning with a common ancestor and their relatedness to one another. So example, we have the cytochrome C protein that's going to be common proteins that are going to be used to determine evolutionary relationships. Because it's evolved slowly with minimal mutations, so it's easy to trace back. Amino acid sequences of cytochrome C in humans and chimpanzees is going to be identical. But there is an 8% difference between human and mice, and a much greater difference between humans and yeast. So here you see the percent differences in that cytochrome C amino acid sequences in different species. Now looking at that one, the rhesus monkey, that's the one that they use to do a lot of different testings on in labs because they are so closely genetically related to us. And that's also the one where we get the Rh factor in our blood typing. It came from first being able to see this protein on the blood cell of the rhesus monkey. So that's how it got the name Rh. Field and laboratory studies are going to provide evidence of evolution through direct observation. So field work is going to be performed in the Galapagos Islands by Peter and Rosemary Grant for 40 years and is going to reveal evolution in action as they have observed and documented several directional shifts in the morphology and populations of finches on the Daphne major due to harsh environmental changes that are occurring on the island. So looking here, we can see the beak depth. You can see how it has changed over time, and so how it has gotten a lot thicker looking and are a lot more broader. They're able to provide evidence of evolution through direct observation. So laboratory studies are going to demonstrate evolution in action as the prevalence of antibiotic resistance that began upon Alexander Fleming's discovery of penicillin in 1928. The isolation and purification of penicillin for mass production in the early 1940s by Howard Florey and Ernest Chain, and the introduction of penicillin as an antibiotic treatment for Allied forces during World War II. As early as 1942, Streptococcus aureus began developing antibiotic resistance to penicillin. And today, more than 90% of S. aureus strains are going to be resistant to antibiotics due to the presence of the enzyme known as pelicinase. Remember, ACE is going to be an enzyme. Enzymes are going to be proteins. So from 1943 to 2010, 12 different antibiotics were introduced. And by 2011, several different bacterial strains were resistant to most of those, if not all, antibiotics. So this has led to MRSA, a deadly bacterial infection. And so this gives you a broad look at when different types of antibiotics were introduced and when the first antibiotic-resistant strain was identified. So penicillin was introduced as a medication in 1941 during World War II, and they started to notice it was antibiotic resistant. The Staph aureus was antibiotic resistant by 1942, so it took about a year. And so you can go down and look at these different things here. So we have combined fieldwork and laboratory studies performed by Colin Donahue, and they revealed that the morphological changes in body size, limb length, and relative toe pad size in anole populations of Turks and Caicos, as well as other anole populations in the Caribbean, in response to high winds that are generated from hurricanes. So you can see there, and you can look at those limbs and see how they've changed. That's going to finish us up for our last unit for Biology 101. Hopefully I'll see you back for Biology 102.